Section 2 of Gray's Anatomy, Part 1. This is a LibriVox recording. All LibriVox recordings are in the public domain. For more information or to volunteer, please visit LibriVox.org. Recording by Ruth Golding. Anatomy of the Human Body, Part 1, by Henry Gray. Embryology. 2. The Ovum. The ova are developed from the primitive germ cells which are embedded in the substance of the ovaries. Each primitive germ cell gives rise, by repeated divisions, to a number of smaller cells termed oogonia, from which the ova or primary oocytes are developed. Human ova are extremely minute, measuring about 0.2 millimetres in diameter, and are enclosed within the egg follicles of the ovaries. As a rule, each follicle contains a single ovum, but sometimes two or more are present. Footnote: see description of the ovary on a future page. End of footnote. By the enlargement and subsequent rupture of a follicle at the surface of the ovary, an ovum is liberated and conveyed by the uterine tube to the cavity of the uterus. Unless it be fertilized, it undergoes no further development and is discharged from the uterus. But if fertilization take place, it is retained within the uterus and is developed into a new being. In appearance and structure, the ovum differs little from an ordinary cell, but distinctive names have been applied to its several parts. Thus, the cell substance is known as the yolk or ooplasm, the nucleus as the germinal vesicle, and the nucleolus as the germinal spot. The ovum is enclosed within a thick, transparent envelope, the zona striata or zona pellucida, adhering to the outer surface of which are several layers of cells derived from those of the follicle, and collectively constituting the corona radiata. Yolk. The yolk comprises 1. The cytoplasm of the ordinary animal cell with its spongioplasm and hyaloplasm. This is frequently termed the formative yolk. 2. The nutritive yolk or deutoplasm, which consists of numerous rounded granules of fatty and albuminoid substances embedded in the cytoplasm. In the mammalian ovum, the nutritive yolk is extremely small in amount, and is of service in nourishing the embryo in the early stages of its development only, whereas in the egg of the bird there is sufficient to supply the chick with nutriment throughout the whole period of incubation. The nutritive yolk not only varies in amount, but in its mode of distribution within the egg. Thus, in some animals it is almost uniformly distributed throughout the cytoplasm. In some it is centrally placed, and is surrounded by the cytoplasm. In others it is accumulated at the lower pole of the ovum, while the cytoplasm occupies the upper pole. A centrosome and centriole are present, and lie in the immediate neighbourhood of the nucleus. Germinal vesicle. The germinal vesicle, or nucleus, is a large spherical body which at first occupies a nearly central position, but becomes eccentric as the growth of the ovum proceeds. Its structure is that of an ordinary cell nucleus, viz., it consists of a reticulum or karyomitome, the meshes of which are filled with karyoplasm, while connected with, or embedded in, the reticulum are a number of chromatin masses or chromosomes, which may present the appearance of a skein, or may assume the form of rods or loops. The nucleus is enclosed by a delicate nuclear membrane, and contains in its interior a well-defined nucleolus or germinal spot. Coverings of the ovum. The zona striata or zona pellucida is a thick membrane which, under the higher powers of the microscope, is seen to be radially striated. It persists for some time after fertilization has occurred, and may serve for protection during the earlier stages of segmentation. It is not yet determined whether the zona striata is a product of the cytoplasm of the ovum or of the cells of the corona radiata, or both. The corona radiata consists of 2 or 3 strata of cells. They are derived from the cells of the follicle, and adhere to the outer surface of the zona striata when the ovum is set free from the follicle. The cells are radially arranged around the zona, those of the innermost layer being columnar in shape. The cells of the corona radiata soon disappear. In some animals they secrete, or are replaced by, a layer of adhesive protein, which may assist in protecting and nourishing the ovum. The phenomena attending the discharge of the ova from the follicles belong more to the ordinary functions of the ovary than to the general subject of embryology, and are therefore described with the anatomy of the ovaries. Footnote: see description of the ovary on a future page. End of footnote. Maturation of the ovum. Before an ovum can be fertilized, it must undergo a process of maturation or ripening. This takes place previous to or immediately after its escape from the follicle, and consists essentially of an unequal subdivision of the ovum, first into 2 and then into 4 cells. 3 of the 4 cells are small, incapable of further development, and are termed polar bodies or polar-cytes, while the 4th is large and constitutes the mature ovum. The process of maturation has not been observed in the human ovum, but has been carefully studied in the ova of some of the lower animals, to which the following description applies. It was pointed out on page 37 that the number of chromosomes found in the nucleus is constant for all the cells in an animal of any given species, and that in man the number is probably 24. This applies not only to the somatic cells, but to the primitive ova and their descendants. For the purpose of illustrating the process of maturation, a species may be taken in which the number of nuclear chromosomes is 4. If an ovum from such be observed at the beginning of the maturation process, it will be seen that the number of its chromosomes is apparently reduced to 2. In reality, however, the number is doubled, since each chromosome consists of 4 granules grouped to form a tetrad. During the metaphase, see page 37, each tetrad divides into 2 dyads, which are equally distributed between the nuclei of the 2 cells formed by the first division of the ovum. One of the cells is almost as large as the original ovum, and is named the secondary oocyte. The other is small, and is termed the first polar body. The secondary oocyte now undergoes subdivision, during which each dyad divides and contributes a single chromosome to the nucleus of each of the 2 resulting cells. This second division is also unequal, producing a large cell, which constitutes the mature ovum, and a small cell, the second polar body. The first polar body frequently divides while the second is being formed, and as a final result 4 cells are produced, viz., the mature ovum and 3 polar bodies, each of which contains 2 chromosomes, i.e. 1/2 the number present in the nuclei of the somatic cells of members of the same species. The nucleus of the mature ovum is termed the female pronucleus. End of section 2. Recording by Ruth Golding.